Good day, this is Professor Will, CBMD PhD, and today is Tuesday, June 29, 2021. It is 5:01 and 39 seconds, p.m. Eastern Standard Time. And today's research, genetic diversity. And don't forget to check out my website. So let's hop to it here. Call to increase genetic diversity in immunogenomics. Date June 29, 2021. Source, University of Louisville. Historically, most large-scale immunogenomic studies, those exploring the association between genes and disease, were conducted with the bias toward individuals of European ancestry. Researchers are now calling to actively diversify the genetic resources immunogenomics researchers use in their work to advance genomic medicine more equitably. As part of an international group of researchers who say the narrow studies limit their ability to identify variation in human adaptive immune responses across populations, researchers need to better understand how genetics influence immune system function by studying population cohorts that better represent the diversity observed across the globe. If we are to fully understand disease, susceptibility as well as design more tailored treatments and preventative measures, researchers analyze diversity in immunogenomics, the value and the challenge the group advocates for resources used in immunogenomics research to actively include and specifically identify additional populations and minority groups. They say such diversity will make their research more relevant and help in understanding population and ancestry-specific gene-associated disease, leading to improvements in patient care. As scientists, we have a say in which populations are investigated. Therefore, it is critical for us to be actively inclusive of individuals representative of the world we live in. This is especially critical for genes that are as diverse and clinically relevant as those that encode antibodies and T-cell receptors. The research focuses on immune function and molecular genetics. The team is studying a specific area of the genetic code that controls antibody function. And to better understand how differences in individuals' genes determine their susceptibility to certain diseases or immune responses to vaccines, the team is conducting the largest sequencing efforts of the antibody gene regions in humans and in animal models. And specifically in humans, researchers are working to build catalogs of genetic variation in samples from multiple ethnic backgrounds, and are engaged in projects that seek to understand how this genetic variation influences the immune response in infection, vaccination, and other disease contexts. Researchers are involved in efforts to improve the resources and data standards for antibody and T-cell receptor genes for immunogenomic researchers around the world. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.